Okay, P-Bio. I'm sorry I can't be there today. We have a sick kiddo at the house, and I got to be here with her. So I'm going to run through what I was going to give you as a little mini lecture today. So if you feel like you would like to do this later and add it to your notes, and you want to work on your prairie plants, by all means do so. But let's just kind of get this stuff finished out. So we're in the photosynthesis packets. We finished out the Calvin cycle, general steps for that. Remember, we did fixation, reduction, and then regeneration. We'll do a final summary for that chart at the end of this little mini lecture. On the backside of that note sheet, it says G3P, and we kind of talked about this very quickly on Friday, but G3P, or I'm sorry, on Thursday, G3P is 1/2 of a glucose molecule, right? So that is the molecule that we're shedding out from one turn of the Calvin cycle will give you one of those G3Ps. So G3, that is standing for the 3 carbons, because remember we took in 3 CO2s when we ran the Calvin cycle. So here's the Calvin cycle, right? And there was Rubisco right there. And we tore out, after we used our NADPH, with our high energy electrons, we're able to pull out a G3P. So that G3P, that 3 carbons that's in this little mini sugar, was the 3 carbons of carbon dioxide that came in here and got energized, right? We use some ATP, use some ATP over here. This is a highly endergonic process to run this whole Calvin cycle. Now, G3P, where have we heard this before? Well, you didn't realize it because I didn't point it out, but G3P, if you took glucose and without the need of oxygen, you start going through and we invest some ATP into this, specifically if we use 2, we run some enzymes in here. We had about 5 steps here, and then we had 5 steps after we split, right, and you end up with pyruvate. As you came along the way, the molecule that is right in here actually was G3P, which is almost pyruvate. So G3P is the molecule that glucose actually kind of got broken up into when we ran through glycolysis. And the beauty of G3P is it serves as an intermediate for all 4 biomolecules. So we can use G3P to make, obviously, carbohydrates, because it is a carbohydrate. But it also serves as a backbone. Well, I should slow down writing here. We could also use as a backbone to build various amino acids. How many? Oh, all 20. So we could use that then to build all of our complex proteins. We can also use it as a backbone for a lot of our fatty acids. In addition to all the fatty acids, we can also use it as a backbone for our nucleotides. So that means that we can make pretty much every single type of biomolecule using those G3Ps. So what am I saying? I'm saying that photosynthesis can help us make life, right? So we're taking CO2 and we're turning it into all these beautiful molecules that run biological systems. So that's what we mean by intermediates, okay? The last piece then in the note says, hey, electrons are the circle of life. You know, the Oomba circle of life, right? That whole Lion King song. I'd do a little dance there for you and hold up Simba / Electron here for you. The electrons, if you remember, started in glucose, right? And then we oxidized glucose, right, which means we lost electrons out of it. Electrons left that glucose and it went into NAD+, which then turned into NADH. And where did that NADH end up going? I hope you can answer. The electron transport chain. And that electron transport chain, we oxidized the NADH, the electron transport chain, with those 4 proteins, 4 proteins that are in the cristae, right? Those took the electrons. So these got reduced, bounced from one, bounced to another, right? We pushed the H pluses into intermembrane space. And eventually, who took that electron? oxygen, right? So oxygen ends up getting reduced, and when it does so, it reacts with H pluses, and now oxygen is no longer oxygen. Oxygen is water. So now the electron is in water, but it's a used electron, so it's a little low in energy. So now here's water that has the electron, right? So this is cellular respiration, top side cellular respiration. We take water and we split it, right? Allowing that oxidation process to happen. And remember, chlorophyll A just got excited by light, lost its electron, so it got oxidized. Well, the electron from water gets fed into chlorophyll A, so chlorophyll A gets reduced, right? It ends up getting that electron, and then obviously water turns into oxygen. It can go back up here, right, and take an electron from the electron transporter chain. With that electron, then another light beam comes in, excites that electron, goes downhill, right, oxidized through the electron transport chain. One gets reduced, oxidized, reduced, oxidized, reduced, and eventually it comes down to photosystem 1, where chlorophyll A is there as well, chlorophyll B. Light comes in, excites that one. This one feeds that system. And eventually the electron goes into NADPH. So now we have the electron in NADPH with it. Then we send over to the Calvin cycle, right? And when we had CO2 coming into the Calvin cycle, ran it through, the NADPH gets oxidized, and the sugar that's in here, a pre-molecule for G3P, ends up getting some electrons, and now we have G3P. So G3P now has the electrons, and if we smash two of those G3Ps together, we have glucose, and around we go. So matter is not created or destroyed, it's just rearranged. So the electron is just getting shuttled around from cellular respiration to photosynthesis and vice versa. Round and round we go, right? So that's the big picture. If you can understand that the electrons are doing this, then your perspective of cellular respiration photosynthesis is pretty spot on, okay? All right, let's kick off the last little piece, kind of the evolution of plants. Plants inhabiting areas that are not normally good for plants or animals for that reason, so they have to have some adaptations to be able to survive those harsh environments. So we look at plant gas exchange first. So if you remember, we kind of talked about plant structure. This is a leaf, so this is a cross-section of a leaf. And we got this upper cuticle and this lower cuticle, and then we got the cells that are what would be of the epidermis. This is the underside of the leaf, but this is the palisades layer, and this is the spongy layer, and then there's xylem and phloem here, and we're allowed to transport water and nutrients through. And remember we talked about how there's these little cells in the bottom of the leaf sometimes that allow gases in and out, right? So we'd bring in CO2 and we release the oxygen. All right, that was the stomata or the stoma, and that allows for that to happen. Now, just like when you and I talk, you can see this really nicely during the cold season. When we talk, we release water into the air. You can see that when you see your breath. It's actually the gaseous water that's in your breath condensing right in front of you, and you're seeing the liquid water. Plants do the same thing. I don't know if you've ever noticed, if you've put like a tarp or a piece of plastic over the grass during the middle of summer, and you lift that piece of plastic up off that grass a couple minutes later, there's all this moisture underneath it. That's from the plants, quote unquote, breathing, right? So they're exchanging those gases and they're letting that water out. So there's a big issue with this, right? If it gets really stinking hot out and the sun is beating down on the plants, and they want to do photosynthesis, they're going to be opening their stomates so that they're getting rid of the oxygen and they're allowing the CO2 in. But the problem is, if they keep doing that, they're going to lose water. The plant's going to start dehydrating, and if it hasn't been a wet time period, we haven't had rain, then as it loses water through this open stomate, it's going to dehydrate more. So what is it going to do? It's going to close its stomates to conserve that water. If it closes the stomates, let's think about what's going to happen to the concentrations of gases. The CO2, which it really, really wants, is going to start going down. and the oxygen that it wants to get rid of is going to start going up. And this poses a big problem. One of the big problems resides in the amazing enzyme that we've already discussed, which is Rubisco, right? So Rubisco is going to have this problem with this change in concentrations that we're starting to see right now in this plant. So normally Rubisco in the Calvin cycle, let's just review, bonds CO2, carbon, into RUBP. Remember RUBP, if this is the cycle here, RUBP was the molecule here, and here's Rubisco, and here comes CO2. Rubisco is going to fix those CO2s onto that molecule that's already in here. That's the RUBP. So CO2 is the optimal substrate, enzyme, substrate, product, right? So we are reduction of RUBP is what we're kind of going after, right? We're trying to get them to gain some electrons as it goes through. And the whole goal is to build sugar, specifically, remember, G3P. So we're trying to rip off that G3P from the cycle and be able to continue this process. But here's the issue. When oxygen concentrations get really high, instead of bonding carbon dioxide, it starts bonding oxygen. Oxygen is competitive substrate. So think of Rubisco as wanting to do the right thing, but when put in the wrong situations, it starts doing bad stuff. And oxygen is nefarious by nature. It's really bad for Rubisco. And instead of actually reducing RUBP, we oxidize. Remember, that means that it loses electrons. So we're actually breaking sugars. That's bad, right? And this is actually called photo, because it's happening in the light, respiration, which means to break things down, right? So this is photorespiration. This is bad because we're not making energy. We're not making high energy molecules. We're using high energy molecules. This is going to decrease productivity, right? Most plants that don't have adaptations for extreme dry, hot areas will belong to a family of plants called the C3s, because it's just this 3-carbon molecule that's feeding this Calvin cycle. So if you have petunias, I'm pretty sure, correct me if I'm wrong, I'm pretty sure petunias are in the C3 family. If you've ever seen petunias, have them out in your yard. They're from a equatorial region. There are some native petunias as well, but they will have issues with really hot environments, and so they wilt out really quickly, as opposed to if you have some coneflower in your yard, or even corn out in the field that might be growing. Those plants are not C3. So there are these other two types of molecules, two types of plants, the C4 plants and the CAM plants. C4 plants, they're going to fix this issue, right, and fix our CO2 in 2 different locations, right? So that's and we'll look at this pattern tomorrow. So 2 different locations. So they are going to do some CO2 compartmentalizing, and then they're going to be able to transfer that CO2 to the Calvin cycle. So they're going to be able to do this 2-location situation. As opposed to C3s, they can't do that. And then CAM plants, this is the easier way to remember it. CAM, C-A-M, the A-M, P-M, that's what they're going to do. So they're going to separate based on time of day. So they're going to be doing a lot of their CO2 incorporation in the nighttime when the stomates can be open, but it's cooler, and then they're going to transfer, and when it becomes daytime, they're going to be able to release that CO2 at the daytime. So one is a location thing, 1, 2, and one is a time of day. I wrote day, sorry. Yeah, time of day situation where we have maybe the sunlight. I said day, but I guess the evening can include that day. So yeah, one is in the evening when things are cooler, and one is going to be during the day. We'll talk through that a little bit tomorrow. All right, so let's finish out. You had your organizer. The only thing that we didn't do yet is the Calvin cycle. So if you have a moment, we do the input, we do the output, we do the enzyme, and we do the location. So input for the Calvin cycle, we're going to use the ATP that came from the light reaction. We're going to use the NADPH, right? So these are the star ins. So you should have arrows coming over from the light reactions feeding this. Hopefully you can draw that in and make that pattern. And then we also have to bring in our CO2, our raw material. And then the ATP, hopefully you're catching the pattern, that's going to turn into ADP. The NADPH is going to turn back into NADP+. And the CO2 is going to turn into G3P. And I'm going to put slash glucose. The major workers here, enzyme-wise, that's our ribisco. Right? We'll also have some dehydrogenases here. And then finally, location. This is all going to happen out in the stroma of the chloroplast. Please try to draw this back to the ADP, going back to where the ADP, sorry, is an input for your light reactions, and then do the same thing for your NADPH, just so you're seeing that it's an exchange, molecules are recycling. But I'll check back on you with this component tomorrow when I see you in class, because we'll be able to fix some things around our household for the kiddo if she's still sick. All right, if you need anything, as always, just email me. I should be available through email pretty much most of the day. I wish you well, and hopefully this makes sense.